Welcome to Chapter 2, Part 2, Control of Microbial Growth. Now in this half we're going to discuss chemical control methods and microbial characteristics. Under the heading of chemical methods, we're going to be discussing how one evaluates a disinfectant. Then we're going to discuss the active ingredients that are found in disinfectants, chemical sterilants, and the like, including phenol and phenolics, halogens, alcohols, heavy metals in their compounds, surface active agents, chemical food preservatives, aldehydes, chemical sterilization, and peroxygens and other forms of oxygen. First, let's start with a question. You're in Walmart or some other store in the area and you're confronted with this. All sorts of cleaners, sanitizers, disinfectants, you name it. How do you pick? Now, as a consumer, most of us pick first on cost and second on how well it cleans, things that we can see, how well it removes dirt and such, and thirdly, and I hate to say this, by whether it stinks or not, how nicely it smells. What we really should be picking it on is how effectively it controls microbial growth, but you have no way of telling that. You can't tell by looking at a surface whether you've sufficiently reduced the number of live microbes or not. So we have testing labs that go through and do this kind of thing. As we go through this portion of the chapter, you're going to be learning about how effective different active ingredients are, and this will enable you to be better able to pick the proper chemical control method for what you're trying to do. Now let's talk about how we evaluate a disinfectant. There are three main tests that are currently common in the United States: the in-use dilution test, diffusion tests, and in-use testing. First off, let's talk about use dilution tests. You're going to be doing one of these in labs, and here we have a picture of a bunch of test tubes. They have decreasing amounts of the disinfectant in the test tubes. Okay. Then what you do is you add the same amount, the same number of microbes, and we generally pick, you can think of it as a rogues gallery of microbes, the ones that cause the most problems clinically, and also those that are hardest to kill. And we look and we see where the tubes become turbid. Okay, right here you can see it's turbid, right here you can see it's not. Right here is the minimum concentration of the disinfectant that has inhibited growth of the microbe. Now at this point we don't know if the active ingredient is killing the microbe or stopping it from growing, and depending upon what you do, doesn't really matter. Okay, so we call this the MIC, the minimum inhibitory concentration. Okay, and this is use dilution. This tells us the smallest dilution, or rather, excuse me, the other way around, the highest dilution that we can use and still inhibit the microbe. For this next one, the diffusion test, we generally use this for antibiotics, but you can use it for surface disinfectants. And since there's a good picture in the book, we're going to use this as an example. Here we put down on agar a lawn of bacteria. Then at the same time we drop a disc that has been soaked in the active ingredient. Then after incubation we see where the organism has been inhibited in its growth. It's kind of like the use dilution test that we looked at previously, okay, where we've got inhibition of growth after incubation in tubes, but in this situation we've got it on a solid surface. And once again we've got the MIC. Right here at the boundary is the minimum inhibitory concentration, because the active ingredient has diffused out of the disc into the agar, and we see where the minimum inhibitory concentration is. Now the only problem is with the diffusion test is that we don't know what that MIC is. You have to go in and take a sample of the agar and figure out how much of the active ingredient is in there. Now with antibiotics, they've done that test and they've figured out what the diameter is for where the MIC is. Okay, and we'll talk about that more when we do the disc diffusion test for antibiotics in lab. But before we move on, there's one thing that I do want to point out. Each of these discs has the same amount of active ingredient on it, but we're looking at 3 different microbes. So you can see here that our gram-negative organism was not particularly sensitive to our phenol here, our hexachlorophene, nor the O-phenylphenol. This gram-negative wasn't sensitive to this one, but it's starting to have some effect here. Over here you can see that our gram-positive is highly sensitive. So if you're looking at the same amount of active ingredient, same agar, same amount of diffusion, and you're looking at 3 different organisms, you can see how sensitive those organisms are to that particular active ingredient. This becomes important later at the end of this chapter when we talk about the resistance of various microbes. It's these kinds of tests that allow us to determine this. And when I say these kinds of tests, I'm including the use dilution tests in it. Now for in-use testing. That's where you take a model area, model operatory. I happen to have a dental operatory here because that's what I worked in for many, many years, testing disinfectants. And that's where you take a model operatory, in this case a dental operatory. It could be a surgical operatory in a hospital. It could be an model exam room that a health clinic would have, whatever the situation is, and you test how well the disinfectant does in a real, unquote, situation. So what you would do is you would take and you would swab the area before you apply the disinfectant, try to pull out how many organisms that you can. Then you would go through the disinfectant, wipe everything down, follow the directions, and then re-swab to see what the reduction in the number of microbes is. Sometimes you would actually put a person in, do a dental procedure, and so you would swab the area after you've had saliva spraying all over the place from the dental drill, and then you would swab before, disinfect, swab after. Now, in-use testing is becoming more and more popular because in the lab everything's perfect. In real life, people don't follow the directions, and they're in a hurry. In active dental offices, they generally have 3 minutes in between patients, and so they're not going to let that disinfectant sit for the recommended 10 minutes. Nobody lets the disinfectant sit on the counters for the recommended 10 minutes. I mean, it's kind of silly to require that, but so this is becoming more popular, and we have a tendency to use it with the use dilution tests, the perfect in-lab testing, so where we know exactly how much of the organism we have, we know what the concentration of the active ingredient is, combining it with real-world testing. Now on to the different active ingredients. Let's start with Phenolics. Okay, I have a bottle of antibacterial hand soap because the active ingredient in that is a phenol. Here's the chemical formula of a phenol. Back at the beginning of learning to use disinfectants, Lister of Listerine fame would use carbolic acid or phenol as a surgical dressing, and also he would spray the wounds before and after surgery of the patients that he was working on, and they had a reduction of sepsis in the patient, of infection in the patient. Well, phenol in highly concentrated form, such as carbolic acid, causes cancer. So we don't use it in its highly concentrated form, and in fact we generally use it in a biphenol form because it's far less toxic. You could drink this stuff in most of its concentration that we use and it's not going to harm you, but it harms microbes. Okay, and because it was phenols and their derivatives were one of the first to be used as a surface disinfectant and as an antiseptic, that we still have a tendency to go back to that as the gold standard. Okay, so triclosan, you want to remember triclosan. Triclosan, if you look at your antibacterial soaps, you'll see triclosan listed. You'll see it listed on garbage can liners that advertise reducing smell. Well, if you decrease microbial growth, you decrease the smell. We put triclosan in all sorts of stuff, everything from baby diapers to cutting boards to shower curtains, you name it. Now the other phenol, or phenolic, that I want you to know is Hibiclens. Its active ingredient is chlorhexidine. It's a I'm not even going to make you learn how to say that. But you've got two phenol derivatives. We've replaced the OH group with a chlorine, and we've got this nice amine containing molecule that hooks it together. Now this means that it's active for longer. It takes the body and the environment longer to break it down, and you'll find chlorhexidine in a lot of antiseptic uses, kind of like triclosan. But with this one, Hibiclens is used as a antiseptic surgical scrub that various clinicians use before they don their gloves. And then also you will find chlorhexidine in oral mouth rinses that patients will use before oral surgery. The microbes in your mouth are your friends until there's going to be cutting to be done, and then you really don't want them in your bloodstream. So you want to decrease the numbers so that your immune system can handle them so that you don't have other infections. Now before we leave phenolics, let's talk about how they act to control microbial growth. They get in and they disrupt cell membranes. Okay, you can see how they would do it. This part is, you got it, hydrophobic, inserts into the cell membrane. You've got your other groups on there that act to pop those cell membranes. So I want you to remember the two examples of phenol or phenolics, and I want you to remember how it acts against cell membranes. On to the halogens. Now remember halogens from the infamous chapter 2. Halogens are the atoms that need just one more electron to have a full valence electron shell. So what do? They go around stealing electrons. Okay, and that wreaks havoc on microbes. Now the most common halogen is chlorine. Now bleach is sodium hypochlorite, and that's the one that you're most familiar with. Not only does it make your clothes whiter, but it kills microbes. We also use chlorine gas in treating city water and swimming pool water, such things. And what happens is that chlorine gets there and it impairs the activity of the enzymes. So basically it shuts down the enzymes function. Bromine, which is one down on the periodic table, it's a little bigger molecule, doesn't evaporate as fast. We have a tendency to use bromine for treating hot tub water. It's hotter, the chlorine would evaporate too fast, so we put bromine in, doesn't evaporate quite as fast. Iodine is also a halogen. It's one down from bromine. Now because it's even bigger, its mode of action is a little bit different. It's still stealing electrons, but what it does is it forms complexes, at least so far as we know. It forms complexes with amino acids, and that inhibits protein synthesis. They also bind to unsaturated fats. Remember, an unsaturated fatty acid is one that doesn't have all of the bonds of a carbon taken up with a hydrogen, so it has some double bonds. So chlorine comes in, steals the electron from the double bond, breaks that double bond basically, straightens out the fatty acid, it's no longer the right shape. So halogens are highly effective in small quantities. Well, at least with bleaches, with your chlorines, there's a problem. And you probably already guessed it. If you're using that for a surface disinfectant, it's going to bleach everything. It's going to bleach your countertops, it's going to bleach your clothing, it's going to bleach your carpets if you spill it, and it doesn't smell very good. So we have a tendency to dilute it out, and we also have a tendency to use other surface disinfectants that aren't quite so caustic. Because, well, by caustic, one thing I forgot to mention, it also steals electrons from your metals. In other words, it corrodes your metals. So we have a tendency to use halogen sparingly or to bind it to other things that kind of reel in its electron stealing capabilities. On to the alcohols. You're familiar with isopropyl alcohol, rubbing alcohol, in its use as an antiseptic, in its use on human skin. Okay, it's highly effective. Not so good as a surface disinfectant. It's really quite good as an antiseptic, not so good as a surface disinfectant. If you take ethanol and use it in high quantities, such as in Lysol disinfectant spray, then that's much better as a surface disinfectant. Alcohols act by disrupting cell membranes. Okay? They also dehydrate cells. So those two things, pulling water out of cells, busting up the cell membrane, is how they kill. Now, one of the problems with alcohol is that it evaporates very quickly. Okay? So with rubbing alcohol, that's not a problem. You're putting it on the skin to reduce the number of microbes, either after you've had a break in the skin or when you're going to put a break in the skin, like for hypodermic injection, etc. You do want it to evaporate quickly. When you're using a surface disinfectant, you don't want it to evaporate quickly. So part of the thing is that we put it in this aerosol can with these bottles of isopropyl. Every time you open it up, you get a little bit of alcohol evaporating, and so your concentration of alcohol to water goes down. So you might start out with 70%, but by the time you're done, you're more like 30%. So we put them in these pressurized canisters so that air doesn't get in as you force it out. And also we put in other chemicals that help the alcohol to lay down so it doesn't beat up on your countertops. And we'll talk more about those surface active ingredients later on. Now let's talk about heavy metals and their compounds. By this we mean salts of heavy metals. Now, by heavy metals, we're talking about lead, which we never use as a surface disinfectant because of its high toxicity. Silver is very good, has low toxicity to humans. Copper can be used, and mercury. Let's talk about mercury. Mercurochrome, you guys remember mercurochrome? Mercurochrome has just a little bit of mercury bound to a protein. And they also toss in some bromine, and I don't know if it's the mercury or the bromine that makes it that nice red, but I remember from my childhood, I just would much rather have mercurochrome on my cuts because it made it look like the cut was much worse. I was competing with older brothers, and so it made it look like you were a bloody mess by the time you got done. It stung, but alcohol stung, and you didn't get the war wound look. Okay, but mercury and other heavy metals bind to the active sites of enzymes and inhibit their use. Now, you're probably going, why in the world would we ever use mercury in anything? Because mercury has gotten a bad rap. It's also, in high quantities, a neurotoxin. But in smaller quantities, generally speaking, it's not a neurotoxin, and it still inhibits the growth of microbes. But because of the brouhaha around mercury in vaccines that originally was linked to autism. By the way, it turns out that the doctor who did that original study didn't use enough subjects, and he was also working for a company that was trying to market their new line of mercury-free pediatric vaccines. He basically falsified his data. So anyway, but we are still very careful with mercury because it is neurotoxin. That's also why we don't use lead. Besides, by the time you get high enough quantities of lead to kill microbes, it's toxic to humans. Now with the others, silver containing compounds, there are silver drops and creams that are put into the eyes of newborns. And this was to prevent gonorrhea transmission during travel through the birth canal, picking it up from the mother basically, getting into the eyes of the newborn, which would later cause blindness. So we would put these silver containing compounds in the eyes of the newborn, very low toxicity, and it would kill the gonorrhea. And it made it so that the babies had little black tears. Also, silver shows up in creams for burn patients, because it's highly effective against Pseudomonas. It's one of your more resistant gram-negative organisms, which is a real problem with your burn patients. And copper is if you're using an anti-algae agent in your decorative pond, it's got copper in it because that inhibits the action of chlorophyll. Now let's talk about surface active ingredients. Soaps and detergents are considered surface active agents. They are, and let's bring up my picture of the soft soap, without the phenolic in it, without the triclosan, the soap itself is still effective as a microbial control agent because the detergent breaks up oils. It also cleans. So it physically removes organisms. Now, you can have a lot of microbes very happily living in your container of soap or on your bar of soap. But when you physically remove them from your surface or from your skin, it's what we call degerming. And this reduces the number of organisms. Okay, so when you've reduced the number of organisms, you still achieve the effect that you want. So we have a tendency to use soaps and detergents in our cleaning agents because in the process of cleaning, we're reducing the organisms. Now let's talk about quaternary ammonium chloride compounds. And this is wet. Here's your ammonium ion. This is basically ammonia that you would use in Windex, and we've attached other stuff to it. This increases how long they're able to be active, decreases the amount of time it takes for it to evaporate, and it makes it so that it lays down very nicely on countertops. Now, we have a tendency not to mix quats, as we call them, with soaps and detergents. The soaps and detergents can inactivate the ions that are going on here. But quaternary ammonium chlorides, quats, are very good at cleaning as well as disinfecting. So you're removing dirt, you're degerming, but also your quats are also popping cell membranes. They're disrupting cell membranes, and they're very effective at that. They have low toxicity to humans, and they have a pleasant smell. So we have a tendency to use quats an awful lot. In food service situations, Clorox wipes do not have chlorine in them unless it says has bleach in it. Lysol wipes do not have alcohol in them. They have quats. They're very good for use in the kitchen. They're very good for surface cleaning when you're going to have food preparation because they have very low toxicity. Now the quaternary ammonium chloride that I want you to remember is Coverage Plus that we've been using in the lab. That is a quat. It's a lovely quat. Now on to chemical food preservatives. Generally speaking, they are a simple organic acid or a salt of an acid. Now if you remember from the first half of this chapter, what do salts do? They pull water out of microbes. They pull water out of the foods. So this, we don't generally put enough in there to dehydrate the food, but we put enough in to slow down the growth of the microbes so that we have enough time from production to consumer buying it to your use that basically we get to eat it before the microbes do. Now remember what acids do? If you lower the pH, that also inhibits the growth of the microbes because most microbes prefer a more neutral pH. So this is kind of the same thing as what we're doing when we salt our hams or we cure our pickles by producing acids in vinegar, or back in the old days getting lactic acid bacteria to produce lactic acid to cure our pickles. They're readily metabolized. We're used to dealing, our bodies are used to dealing with acids and salts, and they're generally judged safe in foods. Now there's been a trend away from chemical food preservatives, and part of this is because nitrates, which are used to cure hams and other processed meats, have been linked to colon cancer. Okay, so it's probably good to eat cured meats in moderation because of the nitrates, and we're also looking at other food preservatives that will be less carcinogenic. But if you decrease the amount of chemical food preservatives, it means that it's got a shorter expiration date, and that generally raises the price. So it's all things that you have to consider. Now up to this point, all of the chemical control methods that we've been talking about have been strictly for disinfection or sanitization. Remember the definitions? Disinfection, you're lowering the total number of your starting number of microbes by 3 logs. You're knocking 3 zeros off of their numbers. Sanitization is anything that's less than 3 log reduction. Now we're moving into chemical disinfectants, or excuse me, chemical control methods that can also sterilize. Let's talk about the aldehydes. Generally speaking, we're talking about glutaraldehyde, but formaldehyde is also an aldehyde. And what is formaldehyde famous for being used for? That's right, is an embalming fluid. That's preserving the body for up to a week or more so that you can have a funeral at a convenient time, so that you have enough time for families to come together and have a funeral before you put the folks into the ground and bury them. This actually started out during the Civil War, when people were concerned about getting the soldiers home to their families instead of burying them far away from home. So formaldehyde. Glutaraldehyde is a little bigger molecule. If you're in the dental industry, you're familiar with glutaraldehyde as a cold sterilant. If you've got instruments or things that can't stand heat or are sensitive to water, you put them into glutaraldehyde and you soak them for up to 8 hours. Generally, you leave it overnight. You may as well. You rinse it off good, and it was sterile when it came out. Unless you rinse it with sterile water, it's not sterile anymore, but you've killed all of the human-acquired microbes of, say, the dental instruments that have been in somebody's mouth, and you don't have the rusting that you would see from putting it into an autoclave. Now, if you use aldehydes for shorter amounts of time, you disinfect. Now, aldehydes are interesting in how they act to control growth, in that they cross-link proteins. They basically freeze them into place. Enzymes, if you remember back to chapter 5, how do enzymes work? They work by moving. They close over the substrate, clip it, or smush them together, and then they open back up and they're ready to go again. Well, if you freeze them into either the closed or the open position, you've killed your enzyme. You can't use it anymore. Say structural proteins that need to move, like flagella. If you freeze them into place, they can't move. Kills the organism. And it's also a good way to preserve them for looking at them under the scanning electron microscope, because you can pull the water out without creating little microbial raisins. Okay, so that's what I want you to remember. Glutaraldehyde is generally what we use. Formaldehyde is also used. Those are your aldehydes. They work by cross-linking, and they have a little higher toxicity. They recommend that you use them in well-ventilated areas because you really don't want to cross-link your lungs. Alright, let's talk about peroxygens. Hydrogen peroxide is the most common form. Okay, they are known as active forms of oxygen. Hydrogen peroxide is H2O2, and generally speaking, in the amount that we use for antisepsis, it's not toxic to humans because you have an enzyme called catalase that breaks hydrogen peroxide, H2O2, into water, H2O, and oxygen, O2. No, that's not a balanced equation, but you get the idea. So that's why when you put hydrogen peroxide onto a cut, it bubbles. If you use it for an oral mouth rinse, you get a mouthful of bubbles. You've got a mouthful of oxygen. So how would this be effective against certain types of microbes? Your oxygen-sensitive microbes are killed by hydrogen peroxide. They don't have the enzymes to deal with it. Okay? And a lot of your pathogens, to a certain extent, are sensitive to oxygen. Also, when you have oxygen wandering around in its active form, it has a tendency to steal electrons. So it acts like your halogens. Steals electrons, disrupts enzyme function, disrupts membranes, all sorts of good stuff. Now, the problem is stabilizing it. Peroxides are not known for being terribly stable in the presence of bioburden. Now, from part 1, remember that bioburden is anything of a biological nature which your disinfectant or your physical control method has to kill through. So, if you're trying to clean up saliva in a dental office, dental operatory, with a hydrogen peroxide containing disinfectant, what's in saliva? Catalase. breaks down the hydrogen peroxide, and it renders it ineffective. Now, recently, within the last 8 years or so, they've been able to stabilize these peroxides, these radical forms of oxygen, so that bioburden doesn't break them down, and they're able to kill on surfaces for a fair amount of time. And we're going to be seeing more of them because peroxygens are actually highly effective. They're nearly as effective in killing microbes as the halogens are. Alright, let's pull it all together. Let's take what we've been talking about as far as active ingredients of chemical disinfectants, physical control methods, and we're going to talk about which groups of organisms each method is most effective with. And we're going to start with the most resistant and go down to the least resistant. Because everything that works with the most resistant is going to work with the least resistant. Alright, prions are next to impossible. Prions are infectious proteins that act by changing the shape of brain proteins from their active functioning shape into an inactive shape. In other words, making them into prions. They stop doing the jobs that they're supposed to do. They just run around making more prions, and the cells die. So you end up with holes in your brain. Mad cow disease is an example of a prion disease, and they're very hard to get rid of. Incineration has been, up until recently, the only thing that you can do. You add heat until you break down the protein until it's amino acids. Makes it inactive. In Europe, they have developed enzymes that will do the same thing. They'll go in and chop up the prions, inactivating them, and we're currently working on getting FDA approval for that for prions. Pretty much that's it. Your next group of highly resistant organisms are your endospores of bacteria, mycobacteria, those are your acid-fast bacteria, and cysts of protozoa. Now, we haven't talked about protozoa much. Cysts are kind of like the endospores of bacteria. They're a resting stage of the protozoa while they're in bad conditions, while they're waiting for conditions to improve. Since they're already waiting, it's hard to kill them. They're already dried out, so you can't dry out endospores and cysts. Mycobacteria are wrapped in a waxy layer, so it's hard to dry them out. They're resistant to heat, they're resistant to radiation, they're resistant to an awful lot of stuff. Things that will work. Okay, with endospores, pretty much you have to go with heat or degerming. And that's the same with all three of these. Now with mycobacteria and the cysts of protozoa, your halogens and high quantities will work to kill them. Ozone, which is an active form of oxygen, will also work to kill them. So if you're mainly with cysts of protozoa, you're worried about your city water supply. With mycobacteria, TB is a prime example. Lysol, if you leave it on long enough, will be effective in disinfecting for mycobacteria. So mainly with these, degerming, chlorines, peroxygens, and in some cases alcohol with a surfactant. Now your next group, your vegetative protozoa, your gram-negative bacteria, fungi including most fungal spores, and viruses without envelopes. These are resistant, but they're less resistant than the previous categories because they're actively living and moving around. So your control methods that disrupt proteins and disrupt membranes are going to be more effective. Okay, but generally your vegetative protozoa, they've got a cell wall that protects them to a certain extent. Gram-negative bacteria, because they've got that outer membrane, that makes it so it's hard for things to get into where it can actually do the job. Fungi have a cell wall. Viruses without envelopes have a hard protein shell. So you have to go with your control methods that act against proteins. So we're talking about heat sterilization, radiation. Desiccation will work to a certain extent, but not as much as you would like. Mainly that's effective against your vegetative protozoa and your gram-negative bacteria. Not so much fungi. Radiation will work. Moving on to your chemical disinfectants, everything that will work against the previous group will also work. Your phenols will work. Your alcohols, if they're in high enough concentration, will work. You don't have to have a surfactant with them. Generally everything except for your quats. Your quats are not terribly effective against this group. Now for the last group, they're fairly easy to kill. So most of your control methods will work against them. So when you look at a product and it says, we kill the flu virus or we kill HIV, don't be terribly impressed. HIV is a virus with a lipid envelope and it's very easy to kill. That's why it's an STD. Has to have fluid-to-fluid contact to effectively get from person to person. It's not built to go through the water system like polio or be sprayed through the air like a cold virus. Okay? So, what do I want you to know from this? I want you to know the 4 basic categories. Prions are in their own group. I want you to know the 3 basic categories that I've outlined here and what groups are effective against them, because on the test, it's guaranteed, I will give you a situation and ask you what kind of disinfectant is most applicable to be used, because it's something that's going to be important no matter what you're going to go on to do in your professional lives or your home lives. You know that you can use a quat in your home, generally speaking, but after a cold has gone through, the quats aren't going to help to decrease those cold viruses. You got to move up the scale. Well, that's it for this chapter. You're probably thinking, enough with the control methods. But like I said, I think this is one of the most important chapters of the whole book. Also, on these summaries, don't worry about what we didn't cover, because I'm not going to ask you about them on the test. But I thought these were nice charts, and they summarize things nicely. If you have questions, feel free to ask me during class, or email me. Feel free to look through this multiple times. Remember, the more times you're exposed to it, the better you're going to remember it.